Hello students, this lecture is about vectors for cloning genes in plants. So the most commonly used vectors for cloning genes in plants is Agrobacterium tumifaciens. Agrobacterium tumifaciens is a soil bacteria which is pathogenic to several dicot plants. It has a plasmid called tumor-inducing plasmid or TI plasmid. It introduces a part of its plasmid called tDNA into plant at the wounded site. So this tDNA has some specific genes. So this tDNA is integrated into the genome of the plant cell. So the genes of the tDNA gets expressed in the plant. They trigger the plant to secrete auxins and cytokines. Already plant is making some auxins and cytokines, and these bacterium, these tDNA genes also influence secretion of more auxins and cytokines. This involves the excess amount of auxins and cytokines that cause undifferentiated cell mass called crown gall tumors in plants. Now, what is the importance of Agrobacterium by infecting the plant? So this tDNA has genes that secrete some unusual amino acid derivative called opines. Bacterium get these opines to the plant. So opines are the source of carbon and nitrogen to the Agrobacterium. So now Agrobacterium is genetically engineered. The tDNA of the Agrobacterium plasmid is taken and is introduced into another plasmid, where in place of the tumor inducing genes, we are adding genes of our interest. Now this modified plasmid is now allowed to infect the plant. Upon infection, as the tumor inducing genes are not present, so there are no tumors caused in the plant, and the desired genes in the plant gets expressed.